Hi, I'm Steve Scherer from the University of Pennsylvania, and I'm here to introduce you to Tom Lloyd, who's from the Johns Hopkins University. In addition to being selected for a special presentation that we're now doing, it's important to note that he also won the Wolfe Prize for Neuropathy Research today. Why don't you tell us something about yourself?
All right, thank you very much, Steve. So I am an assistant professor in neurology and neuroscience at Johns Hopkins. So I mostly focus clinically in neuromuscular disorders, in particular inherited neuropathies. And in my lab, I'm interested in looking at fruit fly models of neuromuscular diseases.
So the poster that you have selected, that we've selected for this year's presentation, do you want to tell us about the background of that?
Sure. So what I've been interested in are these mutations that affect a motor protein subunit, which is present in all neurons. And what is extremely interesting is that mutations in the same domain of the same protein actually cause two very different neurodegenerative diseases. So one that affects motor neurons, and another that is more like Parkinson's disease. And so what we've been interested in is trying to understand why that is.
And your studies on these two diseases, how did you do that?
So what we did is introduce these mutations into Drosophila and looked at their phenotype using a number of different assays. And what we found is that this motor complex, which has long been known to be essential in retrograde axonal transport in all neurons, and is highly conserved from man down into simple organisms such as fruit flies. So this subunit, in addition to being required for retrograde transport along the axon, is also essential for the initiation of retrograde transport at the distalmost end of axons at the synaptic terminal. And what was interesting is that the mutations which cause the motor neuron disease will specifically disrupt that function and not transport along the axon, whereas we don't see that same phenotype in mutations which cause this other neurodegenerative disease known as Perry syndrome.
So that's interesting. So we've known that retrograde axonal transport has been implicated for a long time in a variety of neuropathies and maybe even some kinds of hereditary spastic parapareses. But now this is an exact molecular demonstration of the precise defect in one of those disorders. So what about the Perry syndrome? Was there anything wrong in your studies with that?
So we're working on that. That's our next step is it's really trying to figure out what it is about these other mutations. One interesting thing is that another group at Penn, Erika Holzbauer, has actually confirmed this finding in mammalian neurons independently, and they actually suggest that the mutations will differentially affect transport along axons.
In the findings that you have from your fly model, are there any sort of implications that you've come up with about how that might lend itself to a therapeutic strategy, knowing what you found?
So, since axonal transport is thought to be generally disrupted in a lot of different neurodegenerative diseases, our findings, I think, really suggest that in addition to simply looking at transport along axons, that in these different diseases, one must also look at how the initial steps of retrograde transport are affected in different diseases.
So you have any plans on anything to do next?
Well, we are, one interesting finding is that in addition to this defect in axonal transport, we also see a very severe defect in actually synaptic physiology. And so what we're doing now is looking at how these mutations affect the actual distribution of vesicles at synaptic terminals, and how a disruption of retrograde transport actually leads to an alteration of neurotransmitter release.
It's wonderful. So I think that this is a great breakthrough for science, that we have a model, and even a whole animal model, of a human disease whose molecular pathogenesis has been now described in some detail. And for those people, and perhaps for many other conditions that are of a similar nature, we can look forward to more investigations towards the treatment.